Hi, this is Mary from OpenHelix with this week's Tip of the Week. Now, if you're watching this tip at some other place, be sure to come back to our blog at blog.openhelix.com for additional details about the item that I'm talking about today, the features of the paper that I thought were interesting, and references and links to all the resources as well. But if you're checking us out on our blog, you can just follow me along right now because I'm going to start talking about the human mitochondrial transcriptome, GBrowse. So I found out about this paper a little while ago. I found this great paper in Cell that describes transcription events in the human mitochondria in a number of different cell types, different tissues and cell types. That was really unprecedented, I thought. It's the first time I've seen that much information come out about this. And so what they found is featured in the highlights here. There's a lot of interesting results based on additional types of RNAs that they were finding. They're looking at different aspects of regulation that were uncharacterized before in detail. And so there's a lot of really interesting things in the data. But one of the coolest things about this paper was because they generated so much data, there's really no way for them to present it in this paper. And they created a whole entire GBrowse installation where you can explore the data yourself. So that's what I'm going to be focusing on today. I'm going to be focusing on, right at the end of their abstract, they provide the URL here for mitochondria, maticlab.com. And that's what we're going to check out right now. So I'm going to go right over to that GBrowse. And so what we have here is an installation of GBrowse that's really specific for this paper. And this is one of the really coolest things about this. It's a really nice demonstration that some of the next generation sequencing data that's coming out is just going to be way too big for these papers. And you're going to have to use the software, and you're going to have to understand how to use the software to evaluate the data, to look through it, and explore it the way you would have done in a paper in the past. So what we're looking at here is a version of GBrowse, one of the 2-point-something versions, and it's organized so that you could start to browse around here right from the beginning. But the most important thing is to know that you have to move to the tab here, select Tracks, to illustrate the features that you want. So one of the things I'm going to do is I'm going to show the coding genes right now. From the Select Tracks tab, I'm going to click Coding Genes, and I click back to Browser, and now what we're going to see is coding genes that are going to be appearing on my browser. And so that's interesting enough. And I can zoom in and zoom out and do all the regular things with other GBrowse. But I'm going to show you a couple of other things too. One of the things that I found interesting was that you could choose to look at different cell types. So I'm going to choose to show some different cell types. So here's HeLa 562. So I'm going to show 3 different cell types with the same kind of information, this DNase 1 profile. Actually, if you hover over here, you can see more about the details of what that data contains. But let's click back to Browser, and we'll have a look. And so there are a lot of similarities here, but there might be interesting differences in some of these as well. And that's the kind of thing that you'll have to do to explore the data that's in this paper. You're going to have to choose the types of data you're interested in. You're going to have to show the differences in certain features. Let's look at the regulatory elements. For example, let's go back to Browser and click and show those. And see, you might find that there are regulatory elements, and these might be interesting to explore in comparison with the data that you find in the different cell types as well, or the different techniques or the different technologies that are used here. So this is one of the really coolest things. Now you can also upload and share your own data. If you had some mitochondrial data, for example, from your lab, you could choose to create a custom track and compare it to their data as well. You can also set preferences for using the browser in a general way too. But the main point that I wanted to illustrate was that you can create a whole browser that's specific for the data that you want to show based on your project using the GBrowse software. And then you can make it available to anyone else who wants to explore this data. And the kind of information here and the level of interaction is not the kind of stuff that you'd be able to get from a traditional publication. So I thought that was one of the really coolest things. Now the data is really cool, but the fact that they've created this outlet for it as well is really, really nice. So that's what I wanted to show today, this GBrowse for the human mitochondrial transcriptome. Be sure to check out their paper and check out their browser and look around. Thanks for your time.